before we get on?
Yeah, I'm trying to remember, because I read this paper before I was going
You
to
try to
travel.
remember what your question.
Yeah, I went to before I went to
Thank
travel.
you.
A lot.
Needing more water.
Oh, you're coming back. Okay.
Yeah, I probably already forgot some details. Yeah, I went to Seattle for Christmas and the New Year,
Nice.
and San Francisco visit my friends, my middle school friends.
Oh, cool.
So middle school in China,
Oh,
and we both came over to here.
Oh, nice.
Yeah, and I just came back on Saturday. Yeah. It's been a long time since the last time I traveling. Yeah.
But I'm so glad you get your card. Finally.
Yeah.
Okay.
Yeah.
Yeah, I went to Seattle, and I haven't been to Seattle. I went to San Francisco. I think it's a very nice place.
Oh, during Christmas?
Not during Christmas, just summertime.
Oh, the weather is pretty nice.
But it's even more interesting is Christmas, huh?
Yeah.
I can imagine.
I think my friends told me that the weather is good all the year.
Yeah.
I'm kind of like jealous.
I know.
Because
I
winter's here.
lived in California for so long.
Yeah, winters here is a little depressed.
It's cold.
It's cold, yeah.
You actually.
And not too much sunlight.
No, not right now.
Yeah, we really need a vitamin D, I guess.
Let's move the institute, huh?
Yeah. I just went through these 4 paper. That's the old one, right?
Yeah, that one I think is a classic. I love that paper.
Yeah, they justify these purine-primidine DNA tracks around the breakpoint.
Yeah, I think that's beautiful. I love that paper because, let me see if I find it.
I remember. Yeah, people at the moment think that the breakpoint only happen in the like transcriptional activated regions, right? And I think the first time justify the DNA structures actually matters.
Well, you don't know at this point. They don't know if it's structured or not, but they see this purine-primidine repeats, and then the breakage point is right here.
Yeah.
See, it's 500 base pairs, and this one is also like very short distance. This is why I wanted to show you, because I will show you what we are measuring. We are measuring these distances from zDNA sites in the mouse that is missing the ZBTB43. And then this is so nicely aligning with this one, that these breakage points match with the zDNA in this mouse. So I think this is very beautiful, because here they have this different myeloid leukemias, if I'm right. So all kinds of
lymphoma, right? I think it's
lymphomas.
Yeah, I think they focus on the T cell receptor locus. But usually it will pair with another T cell receptor locus, the translocations. But like these three locations they focus on is that one is from TCR locus, but the other one is not from TCR locus.
It went to the wrong place, right?
And it's not transcriptionally activated. So they think there might be something else to cause the translocation, and they find out that it might be the DNA form.
Yeah. So I think this was maybe the very first paper that would give this idea.
Yeah, I think
that
so.
The breakage may depend on the zDNA.
Yeah.
But here they don't know if there is zDNA in those cells which break. So this is where we are coming in, because now we know this zDNA is there, and the zDNA is not there in the wild type, but it's in the mutant. So we can compare those things, right?
Yeah.
So
Yeah.
this is why I think this is maybe somehow connecting to what you have been doing, right, with
Yeah.
this translocation works, right? And I have a question to you. Did you guys ever look for the zDNA sequences around those translocations that you're finding?
No. No.
Can you look?
Yeah, I can look.
Maybe that would be a nice thing. Like, what is the likelihood of finding one and what's the reality, right?
You mean look at the pattern, right? The
Yes, so-
purine and pyrimidine
Yes, yeah,
repeat.
so these kind of sequences, and that's, so these are the non-canonicals. One is the, I sent you another paper which shows you zDNA is one of them, the G4 quadruplex is another
Oh, zDNA
one.
is one of the G4?
One
you
of
said?
the non-BDNA.
Okay. Oh, one of the non-B form
And then
DNA.
this paper says this purine-pyrimidine base repeats are close to the breakage point.
Yeah,
And
I
this is
noticed that.
zDNA,
Yeah.
you know, but I can imagine that also G4s, G4 quadruplex or other
Another form of a non-B
non-BDNA
form. Yeah.
might be also fragile.
Yeah.
I think now it looks like the G4 is also fragile.
Yeah.
So this you could look
Yeah, I can
if
look.
you are curious about it. I would be curious because that would really justify what I am planning to do on the on those pediatric
Yeah.
disease. If really they would be triggered.
Yeah,
by
we
some
can
break.
screen those patterns on all those translocation already being.
You know all those
documented.
translocations where they are, and then you would just search
Yeah.
around them.
Yeah. it
Like,
should
is
be
there
a-
something in the 2 kilobase distance? Because this everything looks like it's very close.
Yeah.
See, so everything what they found is like few hundred base pairs or a half a kilo base, and then I send you the stickleback fish that was about 2 kilo base. Right, that was very interesting paper too.
Yeah.
That's like in the evolution. You
Yeah,
can
that
see what happened to
paper-
that fish.
That paper is concise. It's pretty short.
Uh,
Right.
yeah.
And
It's
it's,
amazing.
it's published on Science.
I know, and
Yeah.
it's really not much is new about what they
Yeah.
publish in it, but just, you know, they make it sound so
So easy.
complete.
Yeah, so
like
complete. Yeah.
whatever they publish is nothing really so much new, but they were lucky. I think they were lucky to put it out. But
Yeah,
when
I'm
you-
trying to figure out why it's.
why it's in science.
Yeah.
I think why
I think-
probably because they already had many papers before that were leading up to this paper, so it's kind of the
It's like a conclusion paper.
yeah, like
It's
kind of
a-
brings things together, but it's missing something because they think it's zDNA because it's there,
Okay.
but they don't know if it's zDNA because they never mapped the zDNA in the fish germ cells,
Oh.
right?
Okay. Yeah.
So this is
That's
how
what we do, right?
that's what we know, right? So we are in a very good position to tell more, right?
Exactly. Yeah.
And then not only germ cells, but if we could do the same thing for you, this is what I'm thinking, the blood progenitors.
Okay.
Because that would be like the other part of the story. So the germ
Yeah.
cells would change evolution, right? It would change the sequence of DNA in the germ cells which are passed on. So then they just keep passed on. But then in the cancer, it's a different situation. It's the somatic cell
Yeah.
which suffers that breakage, right? And the genetic alteration because of the double strand break. And then if it happens like one hit, another hit, right, which you see, then you can get the cancer.
Yeah.
So I think it's all the same thing, and I think zDNA may be doing the same thing.
Mm.
And I just have a feeling that these ZBTBs are doing something. You saw I sent you one paper which shows how many ZBTBs are so important in the blood.
Hematopoiesis.
Yeah, hematopoiesis.
Yeah, I saw that one.
And then people
it's a
find
lot.
something. Oh, this is important for this, or this is important for that. But nobody actually looked at them from this point. Are they changing any structures, DNA structures? So I
Yeah.
think that could be a broad direction
Yeah.
to go to. I haven't figured out quite exactly what you would be doing, because I also would like to wait for this data from, if you want to look at the
Yeah, I think
translocations.
that's my first to do things to look at these patterns.
If it's really worth going in that direction, but because of this paper, I have a feeling maybe you will find that also in
Okay.
these translocations.
Okay. Yeah.
If you do find it, then it's definitely worth looking into those hematopoietic stem cells and look for the structures.
Okay.
And I think those can be sorted out also. There are so many markers for those cells. We just have to figure out what is the population we are curious about. And then you can either pick some of those ZBTBs, some of them, and test them, or you can think about more like a screening approach.
Okay.
And I'm kind of leaning to that, because then you can make a tester line, and then you cross it with the different mutations, and then you see what you get. But that's a lot of work, bench work.
Yeah.
So this is why I'm thinking I should double check with you what you really want to do, because I know you have the aspiration of become PI, but you can become PI in different ways. You know, you can be a bioinformatics PI, or you can be more like a like a complex PI who does all kinds of things. So I can imagine if you would be curious into this, curious figuring this out, I think then you have a lot of opportunities to either screen or test. You just have to maybe figure out how much you want to be involved, in the lab work.
Yeah.
Definitely we can give you tons of bioinformatics, so you don't need to do lab work if you don't.
I think the work is necessary at some point.
You feel like you wanna?
Yeah.
Yeah.
It's I'm willing to
You're willing to
participate.
get in there.
Yeah.
And then you can shoot for something more, focused. You pick one of those proteins, ZBTBs, and see what if they do it really in those germ cells, right?
Okay.
In the hematopoietic stem cells, or you can go for the screening. And this is why I sent you this double strand break repair. Today I sent you those papers.
Yeah.
They just give you idea. Nothing is perfect. And this is the other thing I would like you to do.
Okay.
To figure out what is the repair mechanism. Repair mechanism for these translocations, or do you also look at deletions in this children's diseases, or with Timoris mostly translocations? This
Mostly are
hematopoietic disease.
translocations.
Nothing like deletion or. Yeah.
Deletion and the inversion. Yeah.
Also inversion.
Yeah.
So then it's worth looking, because there are two ends which are joined together.
Yep.
And sometimes the repair is based on homology, but sometimes there is no way to accomplish homology-based repair, or there is just not enough enzymes to carry it out, or something, and then things can just join together like they were before. That's fine. But they can also miss a few bases.
Yeah.
And then when you repair such way, then you cause the deletion.
Yeah.
And these are and maybe I sent you on repair too, no, didn't I? The repairs, different mechanisms. Maybe I haven't sent it to you. How the repairs work. Maybe this is in the today's batch of papers.
Yeah.
So I think I sent you today that one of those papers explains what happens if this joins together, what happens if it's based on homology, what if it's non-homology based, and error from different mechanisms, right? So if you look at the ends in your inversions or translocations or whatever, you can maybe figure out if they have microhomologies, because I am thinking it is microhomology based repair. And
Oh,
they are
y-
less than 10,
oh, okay.
20 base pairs, maybe even sometimes just 2 or 3 base pairs,
Oh, okay.
right?
You mean like look at the sequence in the patients around the
Yes.
breakpoint to
Yes,
figure
yeah,
out
the
whether
breakage
it's
point in your patients.
like figure out what kind of a
Figure out
repair.
what happened. How was it repaired?
Yeah, I can do that.
Because if you have so much data,
Yeah.
you can
We
maybe,
even have a long read data for the 2 cell lines. I did.
yeah, maybe in those ones you can do.
That's the fused ERG fusion in AML, one of the
Uh-huh.
AML subtypes.
And
Yeah.
this is something you could do in our data too, because we
Yeah.
have a lot of mutations. And it would be good to know how they got repaired. So I'm just like eyeballing, and I can see microhomology.
Okay. We can tell from the okay.
Yeah, I mean, but it takes for a long time, but you
Okay.
can do it much faster.
Yeah.
Now, let me show you that paper.
Yeah, I did a long read, RNA long read
Double strands.
for some AML cell line to figure out the exact sequence at the breakpoint.
No, maybe not this one. And then the zebrafish, I sent you something on zebrafish, because zebrafish has a very nice hematopoietic system,
Yeah.
and the genetics can be done in zebrafish initially, so it would be faster than in the mouse.
Okay.
So we can have a reporter fish, which would go green in case we get this kind of event.
Okay. Yeah.
And then these papers give you a lot of possibilities, but nothing is perfect. But I think we can figure, once we know from your data what kind of repair exists, then we go into that way, and we will make the reporter based on that knowledge. And then, yeah, so this one, I forgot which is the paper. I think it's one of the, if not in here, then this is a mouse. Yeah, maybe this one. No. Sorry, I also read them quite a while ago, so I forgot. I think it's this one. No. Oh, I think maybe this is a whole book. Sorry, and I need to find the gun. Yeah, here this one, chapter 27. Okay, so they have reporters for single-strand annealing, alternative end joining, homologous recombination. Okay, but this is not the best one either. Okay, maybe I have to find you that paper. Okay, here, this is maybe a better one. Let me make it bigger. Okay, so double strand break, and then sometimes it just closes back, or there is this end resection. This is microhomology based repair, so you would find that some sequence here would anneal with that one, and after resection they would join together, and then they could be ligated back. So I have a feeling this is what's working for us in the mouse, in the germ cells, and that's the homology based repair, and this is the single strand annealing. And this is also similar to this, but this needs a little longer stretch of homology, but it's not like its own other allele. Like here, you would get the other allele from the same individual, and then you get a perfect repair. But here, you don't take the other allele, you just take something else from the same chromosome, which looks a little similar. That's the single strand annealing. And then this require different proteins, see? So whatever, sometimes you don't have a protein like this, then this cannot work in those cells, or sometimes you don't have this, like a ROD52 mutant cannot carry this out. So then you kind of push towards the other possibilities, you know.
Yeah, that makes
So
sense.
they can shift.
Yeah.
Sometime you can make one work better because one is missing. So then they have this reporter. So I am thinking we will use one of something like that, kind of a fluorescent reporter. So then we can actually identify such events in the individual fish or whatever in the mouse, and then we cross this mouse with a mutant, like a ZBTB mutant, and then that mutant would not remove zDNA, right?
Yeah.
And then that would break more, so you get more of these green germ cells or green hematopoietic cells, right? So it's you need two ingredients. One is the reporter, one is the mutant. So basically that you would need to build first.
Okay.
But I think the fish has a lot of mutants already available, which we could just buy. One time I talked with Stephanie Granger, he said he can inject those things and we can get a mutant very quickly. Like
So you can see the like DNA break and repair in the real time, right?
Yeah, so and then you could just measure it based on the fluorescence.
Oh,
like you can get many cells which have this. Then you know something is really breaking and repairing, and you know which way it's repairing because this reporter would be specifically
for each repair
you,
system.
yes, but how it's getting repaired. So I think it would be very beautiful. It would be a very beautiful screening strategy. But some things I don't know yet, like what is wrong with those hematopoietic cells and whether they are really triggered by anything nearby, you know.
Yeah.
So you can look for that.
Sure.
And then you can figure out maybe if any of this mechanism is more likely to happen, then you know how to build your reporter.
Yep.
This is what I'm thinking for your big project.
Sure. Yeah, that's quite.
So you can
promising.
read these papers. They are very interesting.
It should be in the new batch you sent me, right?
Yeah, this is the stuff I sent you today. Yeah, and then this is very cute. This spectrum, that just has like all kinds of colors. So if you have one kind of repair, and then the cell will get blue, and then if another repair is green, and the other is yellow. So that's kind of really neat.
Sure.
It's also not perfect for our purpose. I forgot why, but so what they do is here that they do a cutting. So the way they test is that they have this very specific restriction endonuclease which cuts only in one time here in the genome. So they induce a double strand break. But what our method will be different will be that we will include here this zDNA sites. So we let that one break. And then that will be a double strand break, and that has to be repaired. So it's a little harder maybe to build it than this one, because here they know exactly this is the restriction size. I cut it here. So this one I think gets cut out maybe. It's a specific enzyme.
Okay.
Or Cas9, they also use Cas9 nowadays to cut it. But used to be some this long cutter enzyme. Yeah, so this is what I read all these papers, but now I also a little bit forgot about them. But I remember I didn't like anything perfectly, but then you see then if you have this kind of color system, then you can just sort whatever cells you wanna sort, and then you have your own markers for the cell type that you are curious about. So once you figure out which hematopoietic stem cell, which stage you are curious about them, then you can shoot for that.
Okay.
And I don't know enough about that part, when in hematopoiesis this happens. Is it like very early in the stem cells, or just any cell, any blood cell can just go crazy. So this is, I'm hoping you will know. Or you tell me where to read it. Or we need to ask Tim. You know, because
yeah, I need to consult him with this problem, this type of questions.
Yeah.
Should be lineage dependent. What cell they work on in this paper? What kind of?
I think this. Oh wait, see. Heck, 293K. Yeah. So this is a more generic thing. But I think if you know what cells in hematopoiesis you are curious about, then there are a lot of good markers, a good bunch of antibodies, you know. Right? So yeah, this one it says U2OS HEC 293. Yeah. I mean, you can start probably building things in some simple cell line.
Yeah.
And then what makes it specific is your deletor, the mutant. Because the mutants, the ZBTB mutants probably are very specific. Many of them are specific to these cell types.
Yeah, I see
So maybe this doesn't have to be specific. This double strand break.
Yeah, I saw you're talking about this, right?
Yeah. This is, see how interesting, how many of them, and this one of them is called. This PSEF. We have the mutant mouse coming in January, because this is also important in the germ cells, spermatogonia. And Brandon is working on this one. This is ZBTB16. ZBTB16. He's gonna work on it in the spermatogonia. But you could do the same thing in the hematopoietic cells with this mouse if you want. Because I think Brandon is not really interested in the hematopoiesis, so that's NK cells. So, and then there's this Pokemon BCL6 maybe, I forgot. They have so many names.
Yeah.
Did I send you the ZBT? Maybe there was, or maybe here they have something like. Oh yeah, if you go a little higher. Oh no, I thought there was something where they said what is what. Or maybe here. Yeah, I think here, yeah. They have the other names.
Yeah.
See? They just have so many synonyms. I can't remember all of them, but there's something like, this is the Pokemon, see? 7A. Pokemon. She would like that. She's a champion of Pokemon.
Okay.
And then Sikaiso, it's another one. 33. Yeah. Okay, so I sent you the grant, right? Did I send you the grant?
Yeah.
That will be reviewed in January. So it was supposed to be reviewed when the government was shut down, and then they postponed it.
Oh, okay.
So now January 20th.
Okay.
I don't know. So I think I put all those names that I was curious about, but I mean, we don't have to restrict ourselves to that. If you are curious about another one, you can pick another one. If you just want to test one, for example. If you want to do a screening, then we can do a bunch of mutants and cross it to that other one, you know, which would be a fluorescent mouse. Fish. Thinking about. Yeah, I think, and you worked with fish, right?
Yep.
Yeah, so then you know how to work with the fish.
Oh,
Zebrafish.
not really.
What did you do?
I didn't work with fish.
You didn't?
It's Lauren. Lauren worked with fish before.
Oh, why did I think so? Somebody. Okay, so you didn't.
Yeah.
But anyway, I think if we collaborate with Stephanie, she can teach us how to do the
Sure.
fish work.
Yeah.
It's probably not very complicated. You just, if you breed the fish, you just put the female and male in the same aquarium.
But like, how hard to not like generate the mutation in
Oh,
fish?
by micro injecting the zygote. So
Oh,
she
that's
can.
it.
Yeah.
Just inject. Like no CRISPR, no.
CRISPR.
Oh, CRISPR.
Yeah,
Okay.
like you CRISPR something out.
Okay.
So she can do that.
Is it much easier, like, compared to cell line mouse model? Like,
Well, I think we can start with cell line just to see if
Yeah.
the colors come up, right?
yeah.
But the thing is that nothing will happen if you destroy ZBTB43 in that cell because it's not there to start with, right?
Okay.
Because this is specific to germ cells. So that's why when it comes to mutants, you need to make sure that mutation is meaningful to the cell.
So we are, you said testing. So by testing, you mean testing if the mutant will cause DNA break and probably activate some of the repair mechanism, right?
So
Is that what we testing?
So the repair mechanism, I think we need to know what kind of repair we can expect in
Okay.
your hematopoietic cells based on what you see in the sequences.
Okay.
And then we build such kind of reporter which would use that kind of repair.
Okay.
So we just think a little ahead, and we should see some green
Yeah.
because we have the ability to repair, but we are not trying to measure repair. This is measure the repair efficiency or anything.
It's
We don't mess up anything in
qualitative.
the repair. We just want to know how many times it breaks, really. But it's
Okay.
harder to measure double strand breaks.
Okay.
I can't imagine a fluorescent reporter that would measure the breaks. I mean, you can always do gamma H2X staining just to start with, but this would show you the end results of what happens if it broke and it got repaired. But why it breaks is because there is the structure, too much of it maybe, like because we don't remove it.
Okay.
So could be the zDNA or G-force. Because the ZBTB is mutant. So maybe I should show you that. So if you have in our system, what happens is that the zDNA becomes bDNA because we have ZBTB43. This is the normal cell. So then we don't have any breaks. But then if you don't have the protein, so this is mutant, you still have Z, and then that causes double strand breaks, and then repaired, and then you can measure it out, measure the end of this repair, so you have a fluorescence. So then mutation, you are just measuring mutation, right?
Yeah.
So then this would be your mutation, and then this fluorescent would be the other component. I didn't draw it very well, sorry.
Well,
I should work on it.
I think I got it.
I should make a better picture for you. I will make a better picture. I think I may have it in this view. Oh no. Just have so many variations of this. Yeah, I don't have it. Yeah. Okay, so So basically, I think it's going to be the microhomology. This is what I see in this case.
Okay.
But in the other ones, because these are different cells, they are hematopoietic cells. Each of them may have different machinery to repair.
Yeah.
So I'm not sure how to build your reporter.
Okay. I will figure that out.
But what is interesting, so why I'm thinking I might be right about these proteins is that this PRZF, which is the Brandon's, this one, so that one in the reproductive system and also immune and hematopoietic and also in skeletal system it is doing something. It's
Phenotype,
a
okay.
very important
Huh.
gene. So this PRZF,
Okay, so
people mapped where it binds in the spermatogonia, and then they found that it binds the G4
okay.
sequence, which is GGG something, GGG something, GGG something, and they just put that into the supplement.
Okay.
They don't even want
They-
to bring that out, but
Okay.
so because it has these binding sites in those specific cells, I'm just thinking, how about if that protein is changing the G into B
Okay.
in those cells? That's what I told Brando, so he's checking that out.
So
So the mice are coming, and also the G4 structure could be a risk in the hematopoietic cells, right?
yeah.
If it's a big G4 that just cannot be maybe transcribed, translated, and then cannot be used as normal DNA, right? So maybe that's causing the breaks. Yeah, I think it is possible. And then why are there so many of these EBTBs in these hematopoietic cells? So apart from all these other things what people are finding, I can imagine that they have these very interesting structures, these zinc fingers, multiple zinc fingers, that maybe they are specialized to see DNA structures.
Yeah, there's a lot of variability on number of zinc finger. Is this how many zinc finger they carry?
See,
There's
there's
a lot
so
of
many of
variable,
them,
yeah.
so they have so many possibilities,
Yeah,
right?
yeah.
And the BTB domain, that's a dimerization domain. So they have the BTB domain and the zinc fingers.
Yeah, BDB domain is for the protein-protein interaction, I think. Yeah. Yeah. How was the paper? Like the mapping.
Mapping. So that's what we have now is we have the phenotype.
Okay.
The mouse is dying, the newborns. So that's the phenotype. And then we are mapping the mutations, and then we mapping the double strand breaks, and then we mapping the zDNA. So I'm trying to have a big paper of all these. But now we have the difficulties. This is zDNA versus pDNA. So G is mapping.
Is this a p-del sequence?
That's the p-del.
Okay.
So G is working on that one. And we got it to work on fibroblasts very beautifully. But then we applied the same conditions to germ cells.
Yeah,
We just chopped up so it was not possible to.
Yeah, I think G tell me that germ cell is difficult.
It's very
to work
difficult,
with,
yeah.
work with.
Yeah. So now we just use
Too many signals, right?
She thinks the DNA might have many single-stranded parts because
it's in germ cell the genome is more activate, right? It's more
we have
accessible.
this broad transcription, so it
Yeah.
might have something to do with it.
Okay.
But we don't understand the broad transcription, but she found that it is important for methylation establishment. Transcription through the imprinted region that we were studying. If you stop that one, then the methylation is not perfectly established in those germ cells.
Okay.
So then maybe that's why, but she did exactly the same thing what she did for the fibroblasts, and then she got much more cut, so the DNA was sheared to smaller pieces. Then you can't map it. So either we would need to figure out the perfect concentration of S1, really titrate it down, or use less permanganate. But it's so hard to get enough germ cells, to measure out all those variables, right? Or the other possibility is we just don't use permanganate and use the S1 cutting, and then see if we still detect any such footprints there.
Okay.
I don't know, do you know this PDASIC? Did she show you or not?
No.
Or should I explain it? So the PDASIC is like S1. So there is so the zDNA would look like this, and then there would be a bubble before it goes to bDNA. There's a single-stranded bubble, just a few base pairs, right? So basically, this is what you can detect if you cut this. Oh, you
this
have
one?
it.
This one. Right?
Yeah,
This one,
so that's the
bubble.
zDNA.
zDNA.
And also you can find bubble in here in the G4. See, you can attack this with S1 nuclease because that cuts single-stranded DNA. So if you can detect these bubbles, based on that you can kind of figure out whether this was Z or B, because this bubble wouldn't be present in bDNA, right? So then what we see is that if we do the sequencing after the S1 cutting, then there is this cutting, and then no cutting. And there's a little bit of elevation here, but basically it's a missing region, because you can't read out of something if you cut it away, right? So you cannot read into that if you cut it with S1. So you end up with this kind of instead of a peak you get like a dip.
Okay.
So Brandon figured out how to map this, and then we can see that if you can align them with a sequence, which is this purine-primidine repeat, and then you see here you have the repeats, and then at the repeats you have this big dip. And then sometimes this works even without permanganate, but the potassium permanganate actually would modify the T's here in the cell, and then it wouldn't be able to annihilate back after you maybe purify the DNA, it would just always be single-stranded. Yeah, we should give you that PDASIC if you want to read it, that paper.
Yeah.
So basically this is where we are, and then now she's collecting germ cells and does it without permanganate, and hopes that now we don't over digest the DNA, but we still will see the strongest cDNA signals. And then we compare the mutant with the wild type. So we were almost there. I thought, we are now, we know the method, we perfect.
This is the only thing that missing.
So this is missing. Double strand break is done. We collected the germ cells,
Okay.
mutant wild type, and the collaborators mapped the double strand breaks. So we see that here is zDNA site. We have the double strand breaks right there, and we have more in the mutant than wild type. So that looks good. And then the mutation that we are struggling with, this is somehow we don't have a good way of comparing the mutant with the wild type. So Brandon is trying to calculate some kind of statistics.
Calculate
The- The mutations.
mutation.
We have the structural variations.
Oh,
So kin call the structural variants.
okay.
And then we can also plot them like, it looks like exactly like this. So if you have zDNA, you have this. And then you have the same thing in the mutant, maybe that's a little lower. But somehow we would need to normalize it, and we would need to figure out if this and this is different statistically. And we are stuck a little bit with this one. But he's working on it, so maybe you you will hear some good news, but maybe we ask you to
Okay.
help. I don't know.
Yeah, I'm happy to help with
Anything calculation,
yeah.
huh? Yeah.
So I guess I feel like maybe this part can help with. Yeah. What do you think?
Yeah, that would be good. And see, this is one thing I keep here. This is something, so the way we set up these mice is that, maybe I have a, so we have this, okay. Okay, so we have the mutant, we have 3 males, and then we have the sperm, because we expected these things happen in the male germline, so sperm should have a lot of mutations, right? And in one of them have the kidney. And then we have the wild type that has sperm and kidney. And then I made a complication on this, so I made this up in a different mouse line. So the father and mother would have SNPs between them. So the F1, which is coming from the mother and the father, right? And we could maybe map recombination
Okay.
in the sperm, and maybe we can see if the recombination was affected by zDNA. So I was very ambitious, I just put too much stuff in one experiment, and that made it very complicated working too. So we have this Black 6 and JF1 different mouse lines, and then he fused the 2 genomes and he aligned it to those genomes. So, and then I was thinking do something like this, like you would find recombinant alleles. You would know, okay, this is in the sperm. You would have like, this would be SNPs, and then this would be the one haplotype and the other haplotype, and then you could find this switchovers, which is a meiotic recombination event.
That's the.
So this is why if you find this in one location a lot, then that could be a hotspot. See?
Yeah. Is it long read or?
Is it long read sequence what we did?
This is long read.
Mm-hmm.
Oh, okay.
Yeah, we did the PacBio.
Oh yeah.
So
then it shouldn't be
we
too
should
hard
be able to do that, right? And see here, this is what they did. Well, this is a hotspot, for example. So if you look at somatic cells, you either find one haplotype or the other haplotype, because they don't do the recombination. Somatic cells like the kidney.
Okay.
But if you look in the sperm, then you find this recombined alleles. So then, if you find that there's a lot of these in certain place, then that's a hotspot. And then we can look for it if in the mutant we have the suppression of the hotspots. Or
Okay.
I should send you this paper. This is this paper, Felicity Jones. So this is what I was thinking, maybe you would be curious to look into. And see this one, this is the stickerback fish, which I sent you the paper about, but I didn't send you this particular study.
Okay.
So what they did, they did this was the marine, this was the freshwater, and they know that there is this inversion in one of them. So if they cross the two fish with each other, then the recombination cannot occur over the inversion, right? Because they cannot align. So it's very beautiful. They show that actually they are not mapping that. See?
Yeah.
So I thought maybe we could catch something like this in our data, but now I am not so sure, because this, like the whole fish, like the whole chromosome, every single copy has this inversion versus this inversion. So if you cross them together, of course every fish, every sperm that you collect will have this problem of non-alignment, right?
Oh, yeah.
But in our case, these are de novo. Many of them are de novo mutations. You know, they occur just in that instant, in that germ cell.
Yeah.
And maybe you don't have many copies of them.
Mm.
But I think maybe something we can do, we can find these kind of hotspots, if how these hotspots are affected. I think we looked at this particular hotspot was not affected, but there are other ones too. So this was just a crazy idea. I made it very complicated, so we had to back cross the mutation into the JF1 mouse. Then we had a homozygous in the JF1. We had a black 6 homozygous, and then we crossed it.
Do you know where's the data store? Like which folder I can find those long read sequence?
Our data.
Like
Our data.
Where in the HPC?
I show you. We have a secondary.
Are you going to meet with King recently? I'm going to meet
King
with
did.
King.
King did it, and he has a folder in our data.
Oh yeah, I know.
Our group folder.
His folder is pretty big. Yeah, a lot of stuff in King's folder.
So this is the. Oh, you already looked, right? In our folder. So this is my secondary.
Yeah, I can
and
access.
it says King.
Okay.
See? And then it says ZBD43 whole genome sequencing, kidney and sperm.
Okay.
This is that folder.
Whole genome sequence, kidney and sperm.
Kidney and sperm. And then here you have the bumps, and he called the non-BDNA sequences there. Hybrid genome. This is what he created, a hybrid MM39 and JF1.
Okay. Reference hybrid.
This is the reference. Then you have the bumps in here. And I think
That is the raw data.
Huh?
That's the raw data, right? In the BAM folder.
I think this is the alignment
aligned.
onto this hybrid genome. Or the raw data. Wait, FastQC? This one? Maybe FastQC.
That's quality control, I think. That's quality control file.
Oh. Oh, then maybe I don't know. So wait, what's raw data here.
Okay.
Cell 1. High fidelity reads, fail reads. BAM. Yeah, does it say something to you?
Yeah, I think that might be the raw data.
This must be it, huh?
Yeah.
And then I think one of them had to be repeated, so this probably that one. Yeah, so this is something, maybe it doesn't have to go into this particular paper, but
Okay.
if you can figure out
Yeah, I will try.
how to look for them.
Yeah.
Now I even forgot how I was looking at this. I could see which allele, and I could see the breakage point almost at one point, but then later I looked in a different way when he already separated this. And the sniffers is where we have the latest.
The toys to call the structural errors, right? Structure.
Yeah, it caused the
Yeah, I used that
structural
one
variant.
before.
And then he did a lot of analysis on that one.
Do we have a manuscript already?
Huh?
Like, do we have the manuscript for this paper written?
No,
Not yet.
no, I have the written part for this one because
Okay.
that's done, and we don't have enough data for this,
Okay.
and I don't have the figures for this.
Okay.
So it's still a long way to go.
Let me try if I can
I-
help with this.
yeah. And I am keeping back this part because I thought if you put everything together, it's very significant, very meaningful, right?
Yeah.
You bring from zDNA to mutation to the breaks and mutation.
Yeah.
Everything would make a lot of sense
I
together.
agree. Yeah.
So I'm just holding it, and
Yeah.
I don't know. But I feel bad because these people did the work already long time ago. But they agree. They are happy to be in a bigger paper.
Yeah.
So
I have a feeling that I might can help with this. But I need to
if you can help with
read the
it.
paper. Yeah.
You think we should put this in?
Yeah,
mean,
I will
if you find something.
Let me go. I cannot guarantee, but I will go. I will.
Yeah,
I think that's my
that would be very nice.
definitely on my to-do list.
Yeah, because then you could help us get this paper out. That would be, and then at the same time, the zDNA. So this is the genomic part, but there is also the epigenetic part that zDNA cannot get methylated.
Oh, yeah,
and then
yeah,
we have the mapping of the whole genome methylation data.
Yeah.
And we can see that really the sperm is different. We can see that when the methylation gets messed up during the fetal germ cell development. So we have a whole developmental set. And we also have chromatin mapping data, ChIP-seq. So I am working on that one with the Canadian collaborators. But also I'm kind of lost and I don't know how to plot it
Okay.
to make the point.
Okay.
So I don't know if you wanna see that one also. We can. But I just didn't bring it. I worked on it at home and I don't have the latest one. But it's this U-Montreal chip. U-Montreal chip. That's also in Keen
Okay.
because for some reason maybe he downloaded it. So we have big wicks and bombs and fascicles. I don't know. Maybe again we don't have the raw data, but I can ask for it. And then I have lots of Excel files because I am an Excel person, so I'm just sorting Excel and
Okay.
making heat maps in it. But of course, it's not the correct way to go. Yeah, so this is something would be really nice to complete in the coming months, and I think this is really a matter of analysis, and this still requires this experiment to work. But G is ready to submit the DNA, I think, this week, so
Okay.
maybe then they map the zDNA, and then
So you said it's methylation.
oh, so that
Yeah.
could be another paper. So this is all the
Oh.
genetics, right? So
Okay.
from zDNA to break to mutation. But then there is another paper, which is the DNA methylation and chromatin. So we can see how the H3K for methylation is affected, how other things affected, and how the DNA methylation.
Okay.
But what I want to see is the dynamics. So what comes first, and how it affects what. And is it before the other one? But I don't have enough data points.
Okay.
And I think even in that study, if we could show when and where this happened, this zDNA, when does it change, when it doesn't change in the germ cells, and then we could relate that to the ChIP-seq and
Yeah.
DNA methylation, then it would be very beautiful too.
Yep.
And maybe that's possible to put everything in a homologous paper, but I don't know if it's necessary. Maybe not. But maybe this data can be used 2 times, like one time here, one time the other one. Or we can just use minimal data here.
All these are from the like the mouse model you're talking about, right?
Everything is from the germ cells of
Gotcha.
the mouse fetus.
Gotcha.
Yeah.
Yeah.
Yeah, I think it's very, nobody has such thing where you could actually follow from a structure to mutation in the organism. And you can actually switch off the structure, like we can do, right? If we have the ZBTB, it switches to B. If we don't have ZBTB43, it stays Z.
Yeah.
So it's like a beautiful switch.
Yeah.
And you can just look for all these things. Yeah, and G is very good, so technically she's very good, so I trust her. She can get this one.
Okay. Yeah.
Yeah,
So-
and this mutation I think I messed up because I made it too complicated. Oh, I should also tell you, so the black 6N is the mutant and the black 6J is the wild type. So that's also something, a mistake was made, and we are finding differences in the genome just because these two black 6 lines departed from each other
Okay.
over 100 generations.
Oh.
And some of the things that I initially thought were because of ZBTB mutation are because of this N, black 6N.
Okay.
So this is the ZBTB mutant.
Gotcha.
Unfortunately, this is a messed up experiment, but I don't know if I can afford repeating it. But and then this is also cross JF1, and this is cross JF1 for the SNPs point, right? So it's way too complicated. So maybe I should invest and just do the simple way and just have the black 6N and the mutant in black 6N, and then we redo the whole thing. Then look at the data. Maybe you look at the data and then you see can it be rescued?
Yeah.
Okay.
I will try
If
my
it can be
best.
written, like you can really convince people, because I think if you know what is because of the N6, you can just filter those out. Then you know everything else is not because of the N6. Yeah.
I see what you mean. Yeah. So yeah, I guess I have 3 things to do. One is look at the zDNA prong sequence from those translocations.
Yeah, that
Or
would be very
patient
nice.
from
That would be for
leukemia.
your future project.
And also figuring out
The
the
repair.
repair type for those locations, for those translocation, and also to see. I'm going to look at the data of this part, right? Yeah.
Yeah. So I can maybe give you more information on that one if you like. Where did I put that? So I was just plotting, for example, distance to zDNA, and this is the deletions what we map, and this was mapped in the kidney, and the green ones are in the wild type, and the pink ones are in the mutant. So it's just a histogram. Really, I'm just plotting how many exist in this distance to this distance. See, and this is in the sperm. But here we have 3 sperm samples. Here just one kidney. So it's not perfect to compare them up like just this. But what I see is the mutant has more. See, more than the wild type. But this one hasn't been normalized yet. So this is what the Brandon is working on, trying to normalize it to total reads. And then one way to do is just histogram, and then he started plotting density plots, see?
Yeah.
And then he was trying to calculate the difference between the two shapes.
Okay.
And then he seemed to be able to find significant difference between mutant and wild type. But for some reason we are not happy with it, because if it's not normalized, that could be the cause, right?
Yeah.
And then this is the G4s, and they are not significantly different. So this is giving me hope, even though it looks a little taller, but the statistics is not there. So ZDNA is different, statistically different, but the G4 is not. So that makes sense, because ZD43 is specific to ZDNA. You know, that wouldn't touch G4 maybe. That's what we think. Yeah, so this is where we are, and we talked with Zach Madai, and then he gave us some ideas how to work with the statistics. So maybe we know something more. So if we can have something like this, then maybe this part will be enough.
Okay.
And especially if you can figure out something about meiosis. If that is giving us a little more. If the mutant is different than the other one.
Yeah. Okay.
Yeah. And if this works, so I'm so scared now. I'm just so hoping.
Finger crossed.
Yeah, I know. It all has to work, and then it will be very nice. Yeah. Yeah, and then we have other stuff which needs analysis, some data which is sitting there forever from a previous postdoc. But I don't want to confuse you with that one now. But if you want, we can talk maybe another day.
Yeah, maybe another day.
So that would also, somebody would need to just sit down and do the analysis, and then we write it.
Yeah. Oh.
Yeah, some of it, Daisy is working on it, but I think she forgot about me. I need to remind her. And then she already went quite a way in this, and then there's even more stuff which nobody ever touched, and it's been mapped like years ago, but I just didn't have the bioinformatician to do it. Yeah. So if it's easy for you, if you're curious,
Okay.
it can be a lot done, even apart from this.
Yeah.
But this would be really maybe the first one to attack. And then those things, your hematopoietic mutations, I think if nobody looked at it, maybe somebody has looked at it already, no? Like what kind of repair could be, or what kind of if they are close to any ZDNA sites or G4s, no? Nobody looked at them.
I'm not
I don't know,
sure.
I haven't looked for those
I'm not
papers,
sure.
but maybe we should read.
For those T cell and B cell, the repair system will be pretty active, right? Because
Active there, huh?
I think so. Because
Uh-huh.
they always making those combination of
Yeah,
this
the
VDJ DNA
Yeah,
sequence.
so they should be.
Yeah.
But maybe they are just
But
overwhelmed because they are doing too many, and then
Yeah.
sometimes it just goes wrong, and then oops, right?
Yeah, I know the VDJ recombinase is not perfect. It's just it's not precise.
Mm-hmm.
It can be some base off.
Mm-hmm.
Yeah, but I didn't know other lineage, like what kind of repair
Mm-hmm.
they intend to use. Yeah, I need to find out.
Maybe if you examine those breakage points,
Mm,
you can see that.
yeah.
So I think what is happening, for example, if there is a deletion, then you would find that this is deleted, and the microhomology is somewhere here, and where would be somewhere in the deleted part or in the next one. Maybe it would be here. Here it would be something, and then this would align. These two would align, so this would loop out, align here. So one part what you are looking for is next to the breakage point in the deletion, or in version 2, I think. So that's how you need to look for it. It's more complicated because it's not on the two pieces which remain, right?
Yeah.
So you can't just compare this with this. But in case of deletion, it would be like here and here. Or if it's inversion, then you just have to think about how that
Yeah.
aligns, right? It's
Let's-
a little complicated.
Let me just draw the sequence first, then we can maybe talk about how to
Yeah.
identify it.
Yeah, I think
Yeah.
I have something where I drew it out, so I should find that slide for you. Okay, what else did I promise you? So the repair. Did I give you a repair paper? No. Repair paper.
It should be in the double strand break
Oh.
paper batch, right?
There was something in this one of the papers. Yeah. Okay. So then.
Yeah.
Yeah, and sequence to show you how I see that sequence. Yeah. I think we will have a lab meeting Friday, but I don't know if you would be allowed to be there. Then you would hear
Okay.
what we have now. But I am just kind of afraid to bring you here and then Megan would say, no, you can't do that.
Nobody gonna know.
Nobody would know.
Yeah.
Yeah, you're welcome if you're not
Yeah.
worried.
Yeah.
Usually we meet 1:30 maybe lately.
1:30.
I think we changed it recently to 1:30. Let me check. Because they brought it up. 1:30. 1:30. Yeah.
Gosh.
Yeah, we usually meet in the fishbowl. Yeah. 1:30, and then 3 o'clock there is the rip, so it's not much time. And then you will have your orientation Monday.
Gosh.
Yeah.
Okay.
Good. So I should give you this one. Oh, I didn't give you everything about the lab projects, but yeah, you will hear it soon. Maybe this is enough now.
Yeah. I'm like,
Too
it's
much.
too much thing in
Sorry,
my
I
hand
didn't
right
want to
now.
give you so much. Anyway, yeah, I'm just doing so many things. Yeah. Okay.
Okay.
Repair sequence. I find that for you. I feel like I said I would give you another thing. I just can't remember. Yeah. So this is kin, and then you find it in whole genome sequence with kidney and sperm.
Where is slides? It's in the same folder.
This one? Maybe not. I think I have a better one than this one.
Okay.
I should give you the newer one. This one you need this last one, right? These few more slides.
Yeah,
from
yeah.
here. Where did I have it? Maybe it's somewhere in here. Or maybe. Let's see. Try to save it and see where it goes. Oh, okay. It's in my folder. Okay. I think you can access that too. It's
Primary folder.
it's in primary, and then lab data. That's where we have the group. See,
Okay.
and then here you are. I also put the same what I sent you today into this one for you,
Okay.
and I can just dump this one to you also. Discuss with Keen. This is not the best one, but I can give it to you here, so you can see that. So I just dumped it over. See.
Okay.
Mm-hmm. Yeah. Let's see, maybe I have a better one. Mm. Most of the stuff is in my home computer. That's why I like to work at home, because I'm I'm just storing everything important. So whenever I'm here, I can't find anything. Okay, so I don't think I have a better one, but I have it at home. So when I go home, I will put it into here for you.
Sure. Sure.
Whatever. So I'm happy you can actually see things there. Okay.
Cool.
Yeah, I'm so glad you came to us, because this is really something like was so missing from my lab. Is the willingness, expertise of analyzing. So yeah, maybe today was a little too much and little confusing, but I think you understand most the major
Yeah, I'm.
directions, and
yeah.
then little details
Yeah.
you can fill in.
Yeah.
Good.
And
Yeah.
go recap those paper you sent it to me. Yeah. Because I almost forgot.
Which one?
Like the those one you sent before the traveling. Yeah.
Oh, I also put those in there, so it's in your folder.
Yeah, everything is in
You
my.
can-
I put in my folder. Yeah.
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 So
Yeah.
I think I'm
great.
good.
Thank you for
Yeah.
coming.
Thanks. Thank you for the time.
Of course.
Yeah. Oh,
Yeah,
maybe.
oh, computer. So whenever
Okay.
you have a chance, you can test that one, and then you can give me specifics of what you want.
Okay. Can you send? Can I figure that out and send you later?
The computer?
Yeah.
We can whenever you want
Okay.
to figure it out.
Sure.
Yeah.
Yeah.
Yeah. So if you like the convenience of having a strong computer, and you don't need to keep
Okay.
putting it on the HPC, maybe because Teams said that's very powerful.
Okay.
But I have no idea. Actually, I have something in one of the grants I wrote what it is. But I bought exactly what he said was good. I think that's what he had at that time, exact same thing he had, and he said
Sure.
he can do a lot.
Sure.
And then you need a monitor like this, right?
I think. Yeah.
So you just specify what you want,
Sure.
and then for this one also just let me know what you need.
Okay.
And then, but you don't need no rush, just whenever you're ready, you
Sure.
figure it out. Then
Yeah.
if you have something to work with, then right now that's good. Whenever you have to give it back to Tim. Or maybe we can ask if we can buy it off from him.
Okay.
Maybe because
I don't like that one. That one is
Oh, you don't like it?
lagging.
Oh, okay.
Basically, I need one that have lot of RAM. You know.
Lot of RAM.
Yeah, because RAM is the like the priority.
Yeah.
Because I usually open a lot of Windows. You know.
Yeah, so we just ask maybe the IT people.
Mm.
But I think we can just specify what we
Yeah,
want.
I don't need a much like computational power, because everything I can do on the HPC, and but I do need a lot of RAM. Oh, yeah, that's the.
Well, you figure out what it is, yeah.
Sure.
But I think that may be good for you actually,
Yeah,
because that has a lot.
I barely run analyze on the laptop, on the local laptop, but I think we should somehow utilize that desktop.
That might be convenient when you are sitting at your desk anyway.
Yeah.
That might be good. But if you want a stronger laptop on top of it, then just need to say what it is you need. So
Okay.
you get that, yeah. I think I have the specifics somewhere, but I don't know, maybe if you look, can you tell by looking at the computer, or should I find the specifics? Because I wrote about it in a grant. I exactly wrote what the RAM and what everything.
Okay. I think I can find out.
You can look and find out from the computer.
Yeah,
It
I
will
will.
tell you, right?
Yeah.
Okay.
Mm.
Yeah.
Yeah, but I think I would did it we're going to do it later. I need to complete some of the tasks regarding the hiring process next Monday. So yeah, Megan just sent me the email.
Okay.
I need to take care of it right
You
now, I
have to do
guess.
a lot of things. I'm surprised. I thought it should be simpler, because you were
Yeah.
here before, huh?
Yeah. But there's a gap,
Uh-huh.
so
Oh, that's why.
things might be a little
Yeah.
complicated.
Okay, so we just had a good chat about your vacation, huh?
Yeah, we have a good.
At least.
we for this 1 hour we're talking about my trip to
Yes,
Seattle
yes.
and San Francisco.